Okay, Chapter 8 is we start talking about microbial genetics. Again, you'll see some similarities between the genetics of the microorganisms and the genetics of the human system as well, and we'll make that comparison as we go along. The definitions of genetics and the gene are basically the same, and again, the comparison with the human system is there, and thus the definitions essentially will be the same. Genetics basically is the study of what genes are, how they carry information, how information is expressed, and how genes are replicated. The gene itself is basically a segment of DNA. It encodes a particular functional product, usually a protein. And again, when we talk about this, the components of microorganisms, we still see the predominant, the most abundant substance in these organisms is protein. Protein are structural units. Proteins make up enzymes. Proteins make up the DNA. Proteins are a major organic structure inside microorganisms, as they are inside the human system. The genome, and these are all basically just definitions that you should be familiar with. Genome refers to all genetic material in the cell. Molecular study of the genome refers to genomics. And then genotype refers to genes of an organism, and phenotype is basically the expression of or the physical characteristics of the genes. So genome, genomics, genotype, phenotype, terms you should be familiar with. Now, chromosomes are cellular structures made up of genes that carry hereditary information. This may be redundant for many of you who have had basic science, especially related to biology. You may have already be familiar with the term chromosome. Genetics basically is the study of how genes carry information, how they are replicated, passed to one generation to the other, how it affects the physical characteristics or presentations in particular organisms. Now, organisms as a whole can refer to the human or the biological system as well. We're here specifically relating to microorganisms for microbiology. A gene basically is a segment of DNA that codes for a functional product, genetic information in a cell referred to as a genome. You've seen previously the base pairing that's associated, the repeating nucleotides. We've talked about adenine, thymine, cytosine, guanine, uracil. Bases occur in a specific complementary base pairs. The hydrogen bonds from which connect the strands of DNA, adenine with thymine, cytosine with guanine. Information in DNA can be transcribed to RNA by a process referred to as transcription, and then this information is in turn translated into a particular protein. Nice little statement there at the very end that kind of ties everything together. Again, information in the DNA transcribed into RNA, transcription process, and then the information in turn translated or read into protein related to translation. Genotypes in an organism's genetic makeup, information that encodes for all the characteristics and potential properties of organisms. Genotype is its gene collection, in essence the DNA. The phenotype refers to actually expressed properties. Physical characteristics is how I like to relate to it. Helps you to associate physical with phenotype. The phenotype is a collection of enzymatic and structural proteins. So DNA and chromosomes is the form of the long double helix. You've probably seen that in movies or on pictures, magazines, whatever. You've seen the double helix structure. Basically looks like a coiled ladder. If you imagine a ladder, twist the ladder, that's the appearance of the chromosomes, except for the steps of the ladder are basically the base pairs. They're bonded together by hydrogen bonds that make the legs of the ladder. Chromosomes take up about 10% of the cell's volume because the DNA is supercoiled by an enzyme called topoisomerase. Didn't make up these names. Topoisomerase 2 or DNA gyrase. And this basically is an enzyme that's responsible for coiling the chromosomal patterns. Now, when we look at this process of transcription and eventually translation, remember in this process the ladder has to be uncoiled. And not only is it coiled, but then it's folded up, because remember the chemical attraction of histones. I think we talked about histones. We talked about histones in here? Okay, I get an anatomy and micro mixed up, because I get these anatomy questions when I come into micro. But basically the ladder is coiled around histones, and that gives it a quaternary type of a structure. And so because of that, there are enzymes that have to come in there and actually uncoil this supercoil structure to be able to get the ladder to lay out for us to be able to identify clearly what the base pairing is. So in DNA replication, you've got two helical strands that basically unravel. They separate from each other at what's referred to as a replication fork. This is where synthesis of new strands will begin. Complementary base pairing, in other words, there has to be specific pairing, specific bases, and specific sequences all along the ladder for this information, for this genetic information to be passed on to offsprings. This transcription process, and then eventually a translation process, has to match up with the original bases, thus yielding this complementary copy of the original DNA. So segments of new nucleotides are joined to form short strands of DNA. This is done by a specific enzyme, DNA polymerase, very important enzyme in this process in DNA replication. Short strands of DNA are then joined in a continuous DNA pattern. This is by the reaction of DNA ligase enzymes. So a number of enzymes are proteins, remember, a number of enzymes are involved in this process. Considered to be a semi-conservative replication process because each new double-stranded DNA molecule has an original strand. One of the strands are new, the other one original strand, thus referred to as a semi-conservative replication. Now you'll see other forms of replication that will occur in microorganisms as well, and we'll get into a little bit of detail about those as well. In bacteria, we see replication begins at the origination, or at the origin of replication. In some cases, we see two replication forks. They start at opposite directions. So as the ligase comes in and separates the legs of the ladder, the DNA polymerase comes in. Excuse me. You may see that on one end of the ladder of the helix, and then you may see it at the opposite end as well. So DNA replication makes very few mistakes, largely to this proofreading capability of the enzymes that are involved in the process, particularly the DNA polymerase. A model that's given in your book basically shows the genetics of E. coli in this process. I'm not going to go into a lot of detail about that. I really just kind of want you to know the general concepts of DNA replication because it's more applicable to multiple organisms instead of focusing on just each individual one. From time to time, I may use an example just as a model to use, but I want you to appreciate this general concept, which is not far from what you've already learned in other courses. Those of you who have had AMP1, you already are familiar with this process. Those of you who are taking AMP1 right now, this is similar information that we've already gone over most recently in your class. Again, there are nucleotides involved, adenine, thymine, cytosine, guanine. There are polymers of these particular types of bases which have similar images. They're just reversed. There are double helix that are associated with particular proteins. There's the backbone, the deoxyribose nucleic acids, deoxyribose phosphate. Strands are basically held together by hydrogen bonds. You need to know that. These are hydrogen bonds that hold the legs of the ladders together. See, some of you got it down now. When I say you need to know that, you just got to, I like that. Strands basically run parallel to each other, referred to anti-parallel actually because of the complementary base pairing. This is a nice illustration out of your book that shows the sequencing of base pairing. It demonstrates to you in the center the replication fork that you can see. Again, this illustration is out of your book. You can basically see how the polymerase will come in and be able to lay down these little copies, usually in little short segments of base pairs, lay down these little segments to have replication takes place. It can happen in two ends, not only from this direction, but can also happen from the opposite direction as well until all the base pairs are laid down. And again, errors don't happen because the polymerase that's involved in this process continually proofreads and ensures that the information is read appropriately. And you can go through each of these illustrations, walks you through the process of replication inside your book. And as I said earlier, DNA replication is a semi-conservative process. You should know that. Now, in the transcription process, a strand of messenger RNA basically is synthesized from genetic information in the DNA. Adenine in the DNA dictates the location of uracil and replaces thymine in RNA. You're probably already familiar with that base substitution. RNA basically substituted with uracil. In DNA, the base sequence reads as follows: adenine, thymine, guanine, cytosine, adenine, thymine. And then in the messenger RNA, because you see uracil substituted, and you should be familiar with uracil substitution in RNA, you see uracil, adenine, cytosine, guanine, guanine, uracil, and adenine as the complementary base pairing. Now, the region of RNA polymerase in the RNA still uses the enzyme, a polymerase, but because it's with the RNA strand, it's referred to as an RNA polymerase. It's needed for synthesis. The importance of polymerases in these reactions is essential. It binds with the DNA. Transcription begins. It begins at a particular site referred to as a promoter site. This is where the replication will start off. Then there's also a terminator site where we'll see RNA polymerase and a newly formed messenger RNA are released from the DNA, signaling the endpoint of the transcription gene. I'll get your question in just a second. DNA is transcribed and it makes messenger RNA, tRNA, and rRNA. Transcription begins when RNA polymerase basically binds to a particular site, a promoter sequence. Transcription proceeds in one direction, but now remember, even though it proceeds in one direction, it just means it can't reverse and come back in an opposite direction. You can see the process occur from both ends of a DNA helix, but remember, strung out, these things are miles long, and so that process, for it to occur in a efficacious manner, needs to occur from both ends of the DNA strand for this processing to be able to complete. Okay. Uracil is the base substitution in RNA. You don't see uracil in DNA, you only see it in RNA. Protein synthesis is basically called translation. Protein synthesis, basically called translation. And in this process, remember, we're creating proteins in this translation process because it translates the language of the nucleic acids. Remember, the sequence of the nucleic acid actually is the template by which the organism develops. This is where we get our physical characteristics, the phenotype, from. And so it translates the language of the nucleic acids and basically gives a language for proteins. The language of messenger RNA is in the form of what's referred to as a codon. This is a group of nucleotides, typically 3 bases in sequence, that codes for a particular amino acid. Remember, amino acids are the building blocks of proteins. There are 64 possible codons. There are only 20 amino acids. Therefore, the amino acids has more than one codon, but this is referred to as a degeneracy type of a code. Now, so there is a codon. Particular sequences of amino acids, nucleic acids, particular sequence of the nucleic acids form the messenger RNA. This gives the particular code or the particular sequence that reads for a particular amino acids. The amino acids basically are what are the building blocks of the protein. 64 possible codons, 64 possible combinations, but with only 20 amino acids, there means there's a redundancy here, a redundancy of the code, a degeneracy of the code. You need to be familiar with that term. Now, this ensures that adequate amounts of proteins are produced for microorganisms and the biological systems as well. There are other particular codes for particular amino acids. Sense codons code for amino acids. Nonsense codons basically signal the end of protein synthesis. So there are specific codes that will turn this process on in a promoter type of an effect, and then there are specific sequences of these nucleic acids that make a codon that will actually turn off the process, referred to as a nonsense or a stop codon that will end the process of producing more proteins or protein synthesis. Now, this is some of the sequences. Again, this is out of your textbook. I'm not going to ask you to give me the sequences of a particular amino acid, so you don't have to spend time memorizing codons for particular types of amino acids. What I do want you to appreciate, though, is just the arrangements and the importance of the complementary base pairing. If one of these base pairs are changed, then it interferes with the entire sequence, and this is how mutation